What about gender? There are dramatic physical changes that occur between boys and girls, as you all know. But when it comes to learning and memory abilities, there are also quite stark differences. Young adult women often exceed same-aged men on tests of language and verbal ability, such as remembering a list or details of a story, as well as some spatial learning, like remembering where objects are located. But men tend to outperform women on navigating through a maze, which is a different type of spatial learning. In one study, men were better at recalling a map of a fictitious town and learning the route, whereas women were better at remembering the position of the landmarks and details of the town. These differences do not pop up until puberty, though. Therefore, girls and boys tend to do just as well in their early years before puberty, when these types of experiments are catered to their age group. Then what might cause these differences? Could it be cultural stereotypes? It might be some of that, but even non-human animals such as rats show the same thing. Adult male rats are usually better than females at remembering what maze arms do not contain any food, and adult female rats are usually better than males at remembering what arms have already been visited that day. These differences happen again with sexual maturity in rats. Between young or immature rats, there is no difference, similar to humans. This suggests that the differences in learning and memory are more likely to reflect differences in sex hormones in males and females. There is a dramatic increase in the release of estrogen in mature males and androgens in adult males. This increase appears to happen at the same time in both groups and affects the brain in different ways between these groups. As an example, estrogen stimulates neuronal growth and synaptic plasticity, as found in studies on rats. But what about those who get older, such as in their 60s and onward? What are their working memory abilities? Well, it turns out that one in-depth longitudinal study titled Seattle Longitudinal Study tested a bit over 6,000 individuals on tasks that included several different types of cognitive ability and then re-evaluated them at a 7-year interval. The study found that there was relatively little change, as you can see here, from ages 20 to 50. But then once you get above that, you can see a great decline. The scores here include many cognitive abilities, so about 60 and beyond there is a dramatic decrease and decline in abilities. It is interesting to note that working memory and the working memory system is usually the last memory system to fully develop, but it is also the first to go and to show defects in later in life. Older individuals are usually less agile than younger adults to adapt behavior in new ways that provides reinforcement. Even classical eye blink conditioning responses start to decline at about age 40. When you get up to 50 or so, it takes older adults about twice as long to learn eye blinking responses compared to younger adults. In a real-life example, elderly individuals were also able to master mental and physical skills to run computers in a computer program, but at a rate much slower than young learners. Older people also made more errors. However, chess and bridge experts actually improve rather than decline with age. Pilots as well. It was found between 40 to 60 have less accidents. We know that semantic memory usually is strengthened by repeated exposure, as we have talked about, and at the same time someone reaches an old age, this is particularly true. Semantic memory has been encoded and retrieved many times in older adults. Some studies suggest that older adults are more likely to also remember positive information, while younger adults tend to process negative information more deeply. One study involved participants viewing a series of positive images, then negative images. Those who were older had greater recall of the positive images, and younger people had greater recall of the negative images. This may be due to age-related changes again in the brain, like the amygdala, that mediate the emotional processing. Also, for older adults, TOT, or tip of the tongue phenomenon, happens frequently and much more frequently that are aged 64 to 75 years, according to the studies done by Sutherland, Watts, Baddeley, and Harris. You can see the differences in working memory for young and older adults, conditioning differences, differences in skill learning, and some differences or difficulties for older adults to form newer episodic and semantic memories compared to those who are young. At the moment of conception, an individual's genes affect and influence brain function. particularly the BDNF gene, on the human chromosome 11, helps to regulate the production of brain-deprived neurotrophic factor, which BDNF stands for. The BDNF is a protein that is essential for the health of neurons. What BDNF is, and what it does, is appears to affect learning and memory abilities by enhancing long-term potentiation. You don't need to know about all the specifics, but you should know that about 1/3 of people will inherit a copy of a different version, which is the MET allele, which will produce a slightly less effective form of BDNF. An allele is a naturally occurring variant of a gene. What you will see here is that those people who have 2 copies of the MET allele, seen here as MET of the BDNF gene, show the worst performance on a test of memory recollection than people carrying 2 copies of the VAL allele, shown here as VAL-VAL. This indicates that the BDNF gene may influence learning and memory. So we talked about how there is an overproduction of neurons, which increases at an amazing speed. Showed that before birth, 250,000 new neurons are added each minute. So that's each minute. This neuronal birth, or the birth of new neurons, is called neurogenesis. However, neurogenesis is not just all throughout the brain. It typically is in specific areas, such as during gestation, meaning between conception and birth, and Purkinje cells develop in the cerebellum. However, this overproduction of neurons in early life and in the womb, the brain goes through a period of reduction of neurons as well. Remember, BDNF is a neurotrophic factor, and when deprived of it, the neuron dies. This natural cell death is called apoptosis. This distinguishes it between cells that die in the body caused by accident or disease. Apoptosis is a natural cell death. When looking at how apoptosis occurs, striking similarities are seen between Darwinian natural selection, because many of the neurons, which were produced or overproduced, were eliminated through the pruning back process. So a bunch of neurons are produced and only some survive. This image here shows us a study that was published containing one dendrite that was photographed over several consecutive days, 8 days. You can see the spines or the protrusions appearing here, and see how they might disappear as well over 8 of these consecutive days. You can also see that some of these spines survived through the entire experiment, where this yellow indicator is here, but that others come and go. Some even lasted less than a single day. This just shows you how malleable, dynamic, and active the brain can be on even a neuronal level. One area, the prefrontal cortex, is going through dramatic and profound changes in adolescence and may not be fully mature until later in teenage years, and may not even fully develop until early 20s. And this is why you may hear some jokes about younger teenagers or adolescents that they may need to start growing their prefrontal cortex, because this is the part of the brain that can regulate conscious behavior, suppress impulses that are inappropriate, think or plan or correct behavior. This is again where a lot of higher thinking is involved. The interesting thing is that male and females have differences in brain organization when compared to each other's. Some brain areas are larger in one sex than in the other. By adulthood, men's brains have about 100 grams and about 4 billion more neurons than women's brains, according to Packenberg and Gunderson. The lateral frontal cortex is larger in women generally, which may describe why women outperform men in working memory tasks. You can see the pink here shows that these areas are usually larger in women, and the blue usually larger in men overall. As you may know, men and women's brains are wired differently, and the visual spatial tasks and areas responsible for that are usually larger in men, as men tend to outperform women on those tasks. But language processing for women is usually better compared to men.